Hello everyone, I am Harshmeet Kaur and I am PhD third year student. So like yesterday you have learned like how to prepare the artificial membranes. So today we'll jump from like artificial to the biological cell. As the title of our workshop, it says cells to synthetic biology, but in lab sessions we'll go in the opposite way, synthetic biology to cells. Okay, so see the basics already, like sir have told you like about the phospholipids and all. I think I don't need to explain that again, so we can jump on that. Like we prepare the vesicles with the usually in our lab with the phospholipids, huh? So these phospholipids, they generate the, when we add water in them, they generate the bilayer. And then with these bilayer, the one of the system we get is the bilayer vesicles, liposomes, which you prepared yesterday. So now if we compare this bilayer with the biological membrane, like there is a like huge difference. Like only what is same is like the lipid backbone is same, but except that there are like large number of proteins in them, and then there are carbohydrates, and like there are many like different types of lipids also. Like suppose like in this one we can see like, so we have only one kind of lipid, but it will like there is large diversity of lipid in the bilayers. So that is now to start with, first like I will give you a little first intro about the cells which we will be using today. Okay, so they are the HEC293 cells. These cells, they are isolated from the kidney of human embryo, and they have the epithelial morphology. So epithelial, you all might be knowing, like it is the lining of the organs and tissues. So, and these cells, they are after isolating, there have been done some gene mutations to make them immortal. So meaning like they can divide continuously, so we can culture them. Like we need to take a very small amount of them, and then we can keep culturing to like produce large number of these cells. And then also like, so these cells, the reproduction time of these cells is like they double in about 24 to 36 hours. Okay, and so this is just one view of the cells. And also like these cells, they have an adherent property, meaning like in whichever flask we culture them, they adhere themselves to the surface. So this is a view of that adhered HEC293 cells. Now to begin with, so usually we freeze the cells. If we want to freeze for a short time, we can freeze them at -80, but for the longer time we need to freeze them in liquid nitrogen, like which is like -196. So now we can't just take it out from that and just start using. So there is some protocol which we need to follow to revive these cells to attain the normal condition. So like we'll see that first. And to begin with, like cell culture, it is a like very tedious thing in terms of like to maintain the control the bacterial infections and all. Even a one bacterial infection or one parameter is up and down, everything goes. So that is why the very first step is to maintain the sterilization. The reason that is why we are not like making you do there. So I'll be showing you some videos, because this cell culture lab has to be isolated, so we can't make many people enter there. So the first step is like sterilization. We have to always on the UV for 15 minutes before starting anything in that, and after that we have to clean this hood with the 70% ethanol, because that 70% ethanol has been reported to like kill the bacterial cells. So that is why with this concentration we like sterilize the whole lab before starting. And then now, so when we are now, these are from the human, these cells. So they have some condition inside our body, so if we want to culture them outside, so we have to provide them all that things. So this is the concentration of the media which we need. That is like, first is this is the RPMI media. This is a ready-made media which we can obtain, and it contains like all the amino acids or some nutrients which the cell may need, all those things. And then in this again we add, this is the fetal bovine serum, 10% of that is also like it is also helps in growth of cell. It is this is a serum from the like cow fetus. So it also provides us with the further nutrients. And then again, like the bacterial infections are very common in that. So to avoid that, we add 1% of antibiotic. So now the very first step for the cell viable is thawing. So we are maintaining them at minus 80 degrees Celsius. So after taking that from minus 80 degrees Celsius, we have to thaw them very fast, because the media inside that in which we freeze, it contains DMSO and it should not penetrate. If we like thaw them slowly, it will go inside and destroy the cells. So we have to thaw them very fast. Then we'll take this kind of like falcon tube, we'll transfer it in that, and then we'll dilute it with the media so that the DMSO gets diluted. Okay, so now after this,
Harshmi, what do you mean by thawing them very fast?
fast means like they're in freeze condition, no? So to thaw them to the normal temperature, to liquid, to bring them to the liquid stage. So like fast means like I have to from frozen to the liquid state, I have to bring them in a very fast.
How do you do that?
See, usually people, see, this is the very, like, I can tell you very basic thing, but usually people do it with a water bath also. So, like, we have this cell culture facility is very new here. So, we don't, if we don't have that, this is the, like, one alternative you can say.
So, water bath, would you keep it at body temperature?
Yeah, ma'am, 37.
37.
37.
Because that's where they would
Yeah.
Okay.
So, we don't have that alternative. So
because you shouldn't heat higher, right?
Yeah, no,
Then
no.
you will destroy
yes,
it.
yes. We do everything with the cells at 37.
So that's why you're doing it with your hand,
Yes.
because that will likely to bring it down.
Yeah, actually, yeah,
Got
right.
it.
So, after that, then after diluting it with the cell culture media, with this composition, then we centrifuge them, because now the cells, right now they are in the liquid phase, so we need to separate them from the DMSO. So for that we centrifuge, and then we form a like a cell pellet inside that. So,
So that will be like the precipitate.
yeah, that will be like the precipitate. Okay, if the video will be visible. Wait. So it will be formed at the bottom, and then like at the time when centrifugation is, here time is also very critical. So at the time when centrifugation is going on, at the very same time we can this was a bit. At the very same time, then before that we can like keep the things ready, and we can add in the culture flask. We can like add the media. Like the number of cells I am using. I usually use 5 ml of total media. So what I do is like 4 ml I'll add already in the flask, culture flask. Okay. So now my cells till that they are ready. So this is the pellets. The white precipitate kind of pellet will be visible. Okay. You can go there otherwise. So this is the kind of pellet it forms, a white shape. So now I'll take out the supernatant from that, and now 4 ml I have already added in my flask, and I want total 5. So just I'll add 1 ml more in this above the pellet, and now then comes again this critical step. Now they have been in like in the pellet form, so they'll be aggregated.
Correct.
So now again it is we have a task, we have to separate them also, but we have not to destroy them also. So when we'll add a media inside that, we have to pipette it properly but smoothly. So this is a very critical step. It can kill your cells also. So after this, then again we'll uniformly spread it in the culture flask. This is a 25 centimeter square culture flask we are using. So we'll uniformly spread it, and then when we observe them under the microscope, so this is how they are visible. I'll show you. Wait a second. This is how they are visible in suspension. Now, like properly round shaped. But they, like yesterday you saw the GPMVs, they might be much bigger in size than this. So these are like smaller than them. But so they are now. So as these cells have adherent properties, so now we are maintaining them at in CO2 incubator at 37 temperature and 5% CO2, and also the like around 85% humidity. Like these are the important conditions. Even any one of them changes, the cells don't survive. Okay. So now this is just when I have revived. This is the picture for that cells. Now when I view them after 24 hours. So this is how they have settled down. They have adhered themselves and they have taken a different morphology. Now, like if you see here, you will not be able to see that round structure. So basically, like the cells have cytoskeleton. Basically they spread out their cytoskeleton and they take a particular morphology to adhere to the surface.
So what's your substrate to
Substrate, actually ma'am, it is a, I have not quoted anything
We are
in
not
that.
quoted anything,
Yeah.
because it's just to the slide.
Yeah. So it is a culture, especially like designed culture flux. They have like it is made in that way so that it is.
What is the point of adding this carbon dioxide then? Because you put it in some percentage of carbon dioxide was already.
It is in. I am not adding it. It is a condition. So carbon dioxide, it is required for the metabolism. You know. So the media which we have, it uses that to decompose some of its components to give some and I don't remember the component exactly now. I forgot. So like it is required for the metabolism of the
to
cell.
provide something which the cell needs.
Basically for the growth of the cells. Like because it requires the same conditions as the inside our body, no? So just for that, like the proper condition of the internal body, we are giving all these conditions. So now, like now in like 2-3 days of these theoretical lectures, like we have seen firstly we're preparing a vesicle film with lipids, then we can like add on more lipids in that, and then we can add on the proteins in that. So, but like till what extent can we go? Like the plasma membrane, what it has, like there are huge number of proteins that, huge number of lipids in that. So like even adding one protein in a membrane is a big, like very difficult thing. So even because we have to take care of that, we need to form the vesicle also, and also the conditions of the protein should not be disrupted, है ना? So protein gets very easily denatured. So the number of proteins that plasma membrane has, that is like right till now at this stage it is not possible. So in future maybe someone might be able to reach that stage. So to properly duplicate. So then how can we if we want to see something like affecting the live cell, like we are doing with artificial membranes, but that is not exactly that what may happen in cells. How to see how plasma membrane reacts in different things. So for that, this was the alternative which nowadays which is becoming popular, is the extracting the vesicles directly from the plasma membrane. So that are called like GPMVs, the giant plasma membrane vesicles. So now these membrane vesicles, they are directly isolated from living cells. So because of which they retain their lipid and protein complexity. So of their parent cell, they have both of the things in that. And also, so now what they lack is they come out of the plasma membrane, but they don't take cytoskeleton with it. They lack cytoskeleton and also some like internal organelles and all. So which I think we want. Like if we want to only study plasma membrane, so we want to leave cytoskeleton and all the things at the side, है And inside they have a cytoplasm. So they take the cytoplasm with that. So they are like the proper like replication of the plasma membrane, like we're just taking out plasma membranes from cells. So like this is a view in the phase contrast. They'll be visible similar to the vesicles, like the artificial vesicles which we prepare, like because in phase contrast we can't resolve their complexity. So now to prepare them, they are anyway complex in nature, but the preparation for them is much easier than to prepare the artificial vesicles. So for that what we need to do is, in here the critical step is the buffer preparation and your health of your cells. So like firstly I've told you, like when we are reviving the cells, that is a very important step. It should get proper. If one of even the step is not done properly, then you will not get proper adherent cells. They'll die and you will not have the system. So that was an important step. And second is now the buffer which we need to prepare. Like today there are number of methods actually we can even extract these GPMVs with some like mechanical methods, like with the micropipettes and all also. But today what we'll be doing is chemical induction methods with the chemical. So the first buffer which it needs is this is the composition like 150 millimolar NaCl, then 2 millimolar CaCl2, 20 millimolar HEPS, maintaining at pH 7.4. Okay, this is our first buffer. So this we can prepare and always keep it at 4. But there is a second which is responsible for the extraction. Actually, is the active acidulation buffer. So this buffer needs to be prepared on the day when we need to prepare the GPMV. So it contains this DTT and the formaldehyde with the inside this acidulation buffer, the buffer which we have already prepared. We'll add these two extra components in them, and then we'll be having active acidulation buffer. So but usually now after this buffer, what we have to further check is like, usually when we prepare GPMs, we see like the confluency of the cells should be around 80 or more. So they'll anyway get extracted in lower also, but the number will not be more if you want a good number. So this is a like good amount of confluency. So now in our experiment what we can do is, so I have arranged to add some cells in the 6 well plates. So we were not having individual things, so I'll give you like 6 of the people can use one plate, but you can individually use in each section. So first we can visualize, we have a cell imager. In that we can visualize and see the confluency. It will not be this much because it is difficult to maintain this so many confluences. So but anyway, we can first visualize how your cells are looking. You can see that, and then afterwards, after adding buffer, then we can see like what comes out. So in the first step, let's see.
So do you add both the buffers together for you? Is there?
I'll tell you that.
Okay.
Then.
And the final vesicle that you get, it's much larger than the cell size?
No, that also I'll
Okay,
show you.
fine.
Will not
Great.
larger. It will be smaller actually.
Okay, it'll be smaller.
Yeah,
Okay.
yeah.
I was just wondering whether you can do some tricks.
Okay, like first maybe I can show you also. So just to have a view how it should be. So these are the vesicles which I extracted. So these which are flowing inside, these have like, they have come out of the cells, and these some which are attached here, they are emerging. Like this is a little larger view. See, this is the emerging vesicle here. So these all are emerging from the cells.
I see.
And these are the like adherent cells. So let's start. These are the adhered cells. Now here if we you will see like this has 46% confidence, in this 46% of the area it is covered. So like now what we can do is, I'll give you like
Adhered means it is dried or
attached in the cell.
So that is, I believe.
so for attaching, it is directly attaching to the substrate whatever they have given, or you
Yeah, whatever they have given, that only, whatever they have given. I have not done anything. Okay, so we can do one thing, like six people can come, hai na? So you can like write your maybe name, initials here. I'll give you a marker, and then you can take one and check what is your confluency, and afterwards we'll add the buffer, and then you can see how it gets extracted. Huh? So equals to 5. You can. I'll give you marker. First you start. Like this is your this. Ma'am, you can.
whatever I am
Do you want marker?
Yeah, this one. You need to move to the other side. Yeah.
You can do 3, 4, 5, 6. You 6 from this, then you can do next. Like we can keep like seeing and writing the name.
So where do you want me to write?
You can write about this.
Okay. So should I just take it off?
Yeah.
That's better. I think it was the face contrast.
Face contrast.
Cool.
You can see
I just
another
wrote
one.
my
or-
own.
Then you can write there.
I was
This is a little lesser. हाँ लेंगे. Just 23%. Yeah.
Just press this to get the confluence.
Hello. हाँ जी हाँ जी.
Please empty.
Empty?
Empty means
नहीं. Like that they look like empty.
they look empty.
हाँ.
You can reuse these things or you can reuse this.
No, we can't. This is a one-time use.
Okay.
Okay, so now this other group can start till that, like we can do the process in that, है ना? So. बीकर लेके आना. Also, you can set for those spaces which is all there. And this card. Fluorescence अंदर वाले हैं ना? हाँ.
18.
नहीं ऑटोमेटिक नहीं कर रहा वो एक बार सेट कर दिया ना. So I can show you this. So what we have to do is I think one by one I will tell you group wise, like you can first see and like sit there so that one group can stand there, है ना? It will be easy.
People with this associated with this person.
So first what we have to do is now in these cells we have the media.
Okay.
Culture media. है ना? So first we'll remove that. Okay. We'll slowly remove that so that the pipette should not touch the bottom because our cells are adhered there.
So this you're just cleaning the pipette, is it?
Yeah, I'm just cleaning.
What is that?
This is a culture media that
That is culture.
हाँ, culture media in which we grow.
Okay.
Which had FBS and all things. So this first we need to remove it.
And your cell pellets are at the...
Yeah, they are at the bottom.
Okay.
Even if we see with the light, like they will be visible, like as a turbid layer, like a kind of a layer. They will be white layer and could be. It is difficult to see.
It is difficult to see.
Yeah, but it is visible. So now the second step is now I'll tell you the role of the buffers. So first buffer which we made, the vesiculation buffer.
Right.
So this is
the one you can make and keep.
Yeah, which we can make and keep, which has NaCl, CaCl2 and HEPAS. Okay. So with this, we'll clean the cells because the all cells will not be live which we have cultured. Some of them will be dead also. So we don't need them, the dead cells or which are not adhered. We need the adhered cells only. So slowly we have to add it so that the cells should not come out. We have not to add with pressure, very slowly. So like this is a little bigger flask, so I'll just wash it with 2 ml of this vesiculation buffer. Slowly ahead. And then I'll just move slowly. Just this much, it's enough, and then I'll remove it. Now we can see here. See this is turbid now. This water like so there are some dead cells or which are not.
So now you're removing what's on top as well.
Yes. So now we are left with only the adhered cells,
Adhered cells.
without any cell debris. So now next step is now we'll have the active vesiculation buffer which had DTT and formaldehyde which we have prepared today itself. Inside the base of that is this only vesiculation buffer, not in water but in vesiculation buffer, DTT plus formaldehyde. So here I am adding that. I'll just take 1 ml. 1 ml is enough for this 25 centimeter square flask. So I'll just add it slowly, uniformly so that it should cover all the surface. And then after this, I have to keep it at 37 degrees Celsius. Now here I don't need the CO2 and all conditions, so I can even use the normal incubator. So we'll be keeping in that the yesterday in which you have used, no? So in that, and then after some time, like when I make it, I usually do it for 10-15 minutes also, but even after half an hour or sometime also, you can keep shaking it so that the cells which are the GPMVs which are coming out, so they can keep getting isolated from the cells. So this you can do to starting. Now you can start.
Yeah, so what do you want to do?
So this is yours, huh?
Yeah.
So firstly you have to remove the media. Like you can, you can you just little less tilts like that so that we don't touch the bottom.
Okay, how do you do it? You just press the thing?
Yeah, I'll tell you.
Yeah.
Just like this. Just press it and then move it inside and slowly take it like that. Don't touch the bottom. Yeah, you can do a little fast also.
So press it, right?
Yeah, press it.
As much as I can remove, right?
Yeah. It's enough. Now you can discard this tip from here.
Right, so just
Press it. Okay.
Okay. Take one, right?
Take one more. Now you can take. Wait, I'll set it for this. I think
1 ml.
1 ml is enough. 1 ml is
Okay,
enough.
so this is how
This
much?
is 1 ml.
Alright, so I just do it again.
Press,
Fully press.
Take 1 more.
Okay.
Now add it slowly. All over. So you can do a little tilt also that it moves. There. Okay. So then slowly we can let it shake it. you can remove this.
Can I hold it in both hands? Yes.
New images is for the image that has been captured. Okay. So while we are targeting the conferences, it is not
Now I discard this.
Yes.
Oh, sorry, I think
No
next
problem.
person can use it. I have to draw a fresh micropipette. So can I just keep it? How
You can give him.
Yeah, you can hold it.
So,
I have to use.
this I am adding like because this is a small part, so my 100 microliters is enough.
Okay.
So you
I
can
can
use.
take it. And this. So again, I press it. It's done.
Yeah. You can spread it all around slowly.
Which one? What you are holding?
No, this is
I
not
just
yours.
gave it to you. I just fixed it by mistake. Yeah, I thought that when they, so it's unused. That's it, right?
Yeah.
Then I just.
Now this will after everyone has added, we'll keep at 37.
Alright.
So
Here you go. Thank you.
वो ना बोल लिया इसमें डिस्कार्ड कर लो टिप्स इसमें हो जाती Okay, so you can see with this, which is one is yours.
see this one.
Now you, pick it up, but slowly that only yours one goes out, not on your else.
I need to.
Yeah, take out, but don't touch the bottom.
Right here.
तुम start करवा दो.
Discard कर.
हाँ कर दो.
Okay.
Now you can discard this tip. इधर discard कर लो हाथ में. Take 1 ml of this.
So it is made up of glass or
Bottom is glass, but above they are fiber.
okay. One.
There is a reading there. We fix it. So I have set it at 1 ml.
Do you have biosafety food then inside?
Yeah, it's this side. अच्छा मैं हाँ तुम इधर देख लो एक बार ठीक है
So discard करना है.
Just float it once.
Okay.
Then now you discard that.
This is the same tip.
Same tip, same tip.
The bottom is.
एक बार तुम ये किसका है फर्स्ट वाला हाँ तुम आ जाओ ये फ्रेश ही है क्या
You have another step, right? Only 2 cards. That micro dip another 500 micro.
So you discard this, right?
Discard करो.
Oh, उसमें बाद उठाना.
नहीं अलग अलग है ना टिप स्ट्रेच कर दिया है माइक्रोमीटर
Okay, तो इसे आप पकड़ना.
वो 500 वाला है 500 वाला से ये इधर ले लो ये है स्मॉल टिप both are same I think both are same you can take this
हेल्थ इंस्टीट्यूट
हाँ वो नीचे इंटरेस्ट करना
जस्ट पुट वन ओके
So वैसे डिस्ट्रीब्यूट करना है
हाँ धीरे धीरे
When pipetting also you can insulate हो गया ना and now take the converting. This is the buffer which we are using for the washing. The first vesiculation buffer. 1 ml ले लेंगे.
we can use that once again just keep the tip like that around we are using same solution right just keep it around
So I will place it right here.
हाँ I think this is your set
Around Kumar.
ये तो
हमें एक एक
हाँ नहीं
हाँ वो मैं तभी रखती first so हाँ ठीक है
तो इसको निकाल ले
इसको खाली कर सकते हैं.
ये 1000 वाला है ना?
This is 1 ml सारे के.
हाँ वन
ओके हाँ अब निकाल रिमूव कर लो अब ऐसे थोड़ा करके नीचे नहीं रख जाना.
I'm trying इतना ही होता है
ओके ओके अब ये टिप फिर रिमूव कर दो. नाउ ऐड ओके ये कितना सेटिंग है 500 कर दो इसको.
$100 वाला want
चलो ठीक है रख.
हाँ
हाँ अब इसको डाल दो 500 है ना. ये this is like ml is like you can add according to the volume. Like bigger one I add 1 ml, this we can add 500. ठीक है हो गया?
निकालना भी है
No, अब निकाल. Now this one, this buffer it contains this plus DTT plus formaldehyde. Formaldehyde they like exert the huge osmotic pressure because of which the vesiculation occurs.
क्यों मत गिरा छोड़ दो
धीरे धीरे से
बस हो गई
जस्ट ये इसको शेक करके धीरे से निकलो पानी वहाँ से
अब फिर से इसी से
हाँ इसी से पूरा डिस्कार्ड कर लो ये टिप को और ये स्मॉल और एक बफर है ना सेलुलर इसको हाँ
ये नया लगा दिया
नहीं 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 if we use the same buffer we can use the tip multiple times unless we touch it
Now
that one inside हाँ
so how much?
ये सेट कर दिया है 500 माइक्रोलीटर जस्ट सबका okay just to interface you just press and take it will take that's it
That's enough.
हाँ that's it धीरे धीरे से complete the surface that's it
Done.
हाँ done you keep it that don't we can use it ये नहीं होता तो चाहिए
ठीक है तो हो जाएगा. Who is the official?
next mine I think PS and down one first
ये से निकलेगा.
एक बार ये चेक कर दो.
हाँ ये
Like it's like it's kind of like you have a cell. Okay. So, like, it's budding, I don't know if you've seen, have you seen GPMVs earlier, like with budding and all?
This is already one
Like, there's a, suppose like this is one GP, like there also if you see the image, the budding of that, like some part of this and it gets separated. So it is the same thing, so like that. So there's a cell, so some part of it will bud and get out. Leaving the cytoskeleton.
Some regions are more can I get that?
Some lipids are more. I've already understand whether is it some area which is preferred for vesiculation is something like that
No,
or is it a random
it's
process?
a random. It's because similarly we give osmotic shock. Maybe you can, if you see our, we have like publication, this year's publication. There you will see some images of the budding. It is there in this system so that there you can see there are different shapes we are getting because of osmotic shock. So that is not like some spot because UV has a uniform spot everywhere. So it is just like where it will feed to orient. Like you have to just vesiculate out from any take on this 1,500. So why now we are using adherents for that is because like they will be at one place. So they can they will be moving in solution, it will be difficult for them because of the number of other components. So when they adhere they spread out their hydrogen. So it will attach kind of
Absolutely amazing. But that, they will. See, have to be super.
Each vesicle, let's say you're studying
You need to make this 500.
what do you use?
this card. Right now we have studied 500 to kind of shapes like how much of
You can use the same
can we
tip also.
tolerate. Like we have compared it with juice. Anyone else can do that of the other, right? So we not study protein. Next further we want to do like there are some enzymes which cleave like the lipid heads and all. They are PLD, phospholipases. So that we want to add enzymes.
It's not uniformly present and present in some part of your membrane. Your population has
That you are right. The all composition will be different for different. Yeah, that we have to do some analysis to like. This is like whenever even if we add protein in juice, it is not uniform. So it is like we see the maximum thing what is happening, like in which maximum population. It's this kind of average. Yeah, there is never same.
Training.
Can you tell me what exactly? Yeah, sure. Like why we are doing. Are you okay? So. We have here these cells. You just saw the image of the cellular derivative structures. Okay, so
So just looking at the PS preparation and extraction.
model system is you can start
These
using
are my big goal. I'll have to go and look up
Out
the
of 30, 70
it will give one and that is what it is difficult to achieve. Like by adding components in the towards best proper type.
So what I understand is that you are trying to get the real, like the living one and then extract the inside one.
Basically, in short, I'm extracting plasma membrane. Okay. So it is a replica of plasma membrane. Okay. So like in this particular process, like so it's taking out what exactly is because I can't see negative. Oh, okay, no problem. So see, in this we have buffers basically. So the reason is basically the main idea is to vesiculate from the cells. How? By the method we're using is giving them osmotic shock. So for that, the first there is a composition they have calibrated. Like this composition, we get the vesiculation. So
When you make osmotic shock,
do you know the osmolarity?
so other
What is osmolarity? Okay, osmolarity is basically like It is the number of moles of something in like number. Suppose I have a 300 millimolar sucrose, so it means I have a 300 moles of like sucrose per liter solution. It's like that. Like it is a number of components. It's concentration. Okay. So whenever we have some system, like suppose the vesicle system also, a normal GUE system also. If suppose I'm preparing it in something like I take 300 millimolar. So it will encapsulate the 300 millimolar inside and outside both it will be there because like yesterday you did a hydration, so that will be common. Now what you do is, if you suppose I'll take sucrose, glucose example, if you like you prepare the GUEs, okay, then from outside maybe you add 350, 400 like higher concentration. So there will be concentration difference, but sucrose and glucose they can't go through the membrane. Like sir has shown today that partially conveyed permeable for them. In terms of size, in terms of because it's more compact now. We are using phosphate. It's there have to be aqueous solution inside and outside. So that is why we form a whole vesicle. So
once they reach the solution में डाल रहे हैं
I'm telling that only. हाँ see. अब हमने ठीक है अंदर we have sucrose, बाहर also we have sucrose. Now we are adding glucose from outside. ठीक है. We'll add to that amount की kind of कि हम like we assume कि बाहर only glucose is left. Sucrose is diluted to large amount. ठीक है. Now no, not neither sucrose can go out and glucose. These are larger molecules. They can't go. Only the water will pass. So because of that, now there is osmolarity difference. Something has to pass to calibrate. So only water is the option that can pass. So basically the water will pass through the membrane, and because of that there will be the changes in the shape because it's a fluid membrane. So there will be changes in the shape. So basically they also, GUEs also, they in that terms we call it budding. It's like they are budding out, like they form these structures. Similarly we are using here for the cells. We are giving them chemicals which will give them osmotic shock. So here it is little more tricky because here we have cytoskeleton also. We have many other things also. So this is a calibrated, concentration. So the first one is we are using, it's a base. Like you say this is a buffer for this for this thing, है ना? So this we are using also to like wash off the cell debris from out, so that our see whenever we wash off the cell debris means the like anything if it is there, we need only adhered cells. We don't need any cell debris or something, so that should not interact when we are doing some experiment. We only need GPMVs later on. So if anything is above that, we want to wash it off and we only want to adhered cells, so that when transformation happens.
Can it is possible कि बाकी सब चीजें रख लो और for other purpose अगर
No. In this system, not because the जो हम use कर रहे हैं, the cells will be dead after that. This is a one-time use only. That is why I have to like give you the small amounts I couldn't give because so this is like the cells are not now.
ये वाला मैं निकाल लूँ?
वो एक हुआ एक और पूरा हुआ है
हाँ बस ये लास्ट होता है इससे this is 500
अब ये वेसिकल्स निकल गए सेल में से, ये है तो साथ में ही ना, सेल्स भी वही हैं.
No. That only I was showing you in that video. अभी तुम अपना बनाओगे तभी तो अपने में देखना.
अब नहीं आएगा उसमें, वो उल्टा हो गया. मतलब 81
that is okay. See, confluence is for always for the numbers you want or for the like good size you want. ऐसा नहीं कि नहीं आएगा, कुछ तो है ना, जीरो नहीं है, दो निकले चाहे तो निकलेंगे. पर निकले चाहिए.
In biological system we are always hopeful.
अरे क्योंकि हाँ इसमें अब क्या होता है ना, like I have
which one is mine and it me there
so like they have like I added around 70%. वो 70 में से 30 ही adhere हुए हैं क्योंकि वो सारे कभी भी नहीं होते है ना. That is the thing. This I am keeping at 57. ये वैसे हम मैं usually I keep for 1.5 hour, but we can always visualize before also. ठीक है, मतलब 1 hour के around तक कर लेंगे visualize कि it is coming out or not.
Is this a very example for which one? This one? Okay, then take another one. And one will this one. Take all? Bubble, can you get any bubble? Then we need to put one everywhere. This will be now and this. One time or like there's some you can add it should be gradual. It's okay. You can discard this one. No, you have to discard this buffer. Like the way you discard. And then this buffer again. This one? Yes. You can fold it like this, so it will be easier for you from doing some you can add 500 like this one. And then add this one. This 500 not take this one. This one, huh? This is the active. Should I wait? इस बफर को तो नहीं चारों this one this got
तो हम रिवाइज कराते हैं क्या होता है ये सबके अलग अलग only one we have to sit here or there subjects
बट ये कि हाँ थोड़ा 4 से पहले ही आ जाना क्योंकि अगर पहले हम एक बार चेक कर लेंगे बिकॉज जल्दी फ्री होएंगे तो जल्दी बचेंगे so we have to go there because 5:30 is starting there
5 से 6 तक पहुंचने बोला है
हाँ
10 किलोमीटर
बोला है बस अभी पर अपने को अभी सेशन के बाद पता चलेगा that it is there or not
incubation just कर रहा हूँ cells growth and divide
they will not divide now
इसको बस करना होगा
थोड़ा टाइम में शेप करना होता है अब मैं कर दूँ
नहीं वो तो कर दोगे आप पूछ लो
हाँ ठीक है रीजन ऑफ टेकिंग हो जाएगी बिकॉज़ वो तो ऑटोमेटिक हो जाएगी बट अटैच ताकि ज़्यादा सेल्स उससे आइसोलेट हो जाए वो अटैच ना रहे दे कम इन सॉल्यूशन उसलिए वी कीप ऑन लाइक
सेल्स न्यूड रह जाते हैं
हाँ और हाँ हाँ या राइट
but getting
yeah
There should be something by here, provided all
bilayer.
But in vesicle form.
Yeah, it is a bilayer, but in vesicle form because we anyway solution outside is aqueous, na, and lipids are coming out. Getting it? It is a bilayer with proteins inside. So like,
Everything inside.
except cytoplasm, it is a complete replica of plasma membrane.
What happens to all these?
Then under
Once I didn't want this solution, writing this cytoplasm. So what happens to the inside and the whatever is there inside?
to make तुम कर लो. हो गया तुम्हारा? ठीक एक कर दो बाकी.
What will the organelle?
That only I was telling you. That only I was telling you there. Nothing only plasma membrane comes. Some part of cytoplasm. This way except leaving all organelles behind, leaving cytoskeleton inside. It's only the plasma membrane that was.
मतलब ये
That is why
मतलब इसको फिर डिस्कार्ड कर रहे हैं फिर you are adding this is the buffer solution right and then you are
Yeah,
extracting
actually that only I was
taking it out again
So,
and then
any other
putting
kind
the
of
active
shape
buffer
is
again
in naturally
taking it out.
like if I don't change anything, like I'm accepting, then I have prone.
So like what does this buffer
If I'm giving a protein sugar, I get a large number.
you take it out
I'll
right? So like
give
this
you
one is
this maybe I'll share with you my paper.
okay you make the debris wash or one
Yeah, sure.
Okay, then we can actually.
You can equilibrate your solution.
Yeah. Just for this.
This is essentially the same as just you have added two components extra.
Yeah.
This is
So
similar.
this is the receptive of this giving it a proper
so the extraction of the cytoplasm that's the thing that's happening under one
Extraction of
That
confused like is it coming that full with the vesicle and inside also it's coming
So
full
if even if we have like
and then you are extracting the inside and keeping the plasma membrane or you are extracting the plasma membrane
interaction of what?
that's what I'm
The confidence area that is where there's very less surface area which is not.
You are asking what will be coming
these
out
membranes will
by
be.
the vaccine.
So you are talking about the membrane of the cell
kind
or
of get is
or
that
the
like
membrane of the vesicles that
since
have
the GUVs
Exactly the
they
plasma
are
membranes.
synthetic
which
ones.
are
Which
which
remains
have
there at the end.
which
in the
remains
real
there.
life systems we have proteins
yeah,
and
yeah.
ion channels in the membrane.
Yeah, they are.
Like
But that is
manufacturing
actually,
that
you're right.
is quite difficult.
If you
So
have to extract
instead of doing that, you are taking
This will take, this will have all the components from
so you take the one which have all the components and then you are so my question is, are you extracting the plasma membrane or are you extracting the thing inside the plasma membrane so that you keep the plasma membrane?
It's not just a joint and
they
come one by one.
So you can
also
So inside the
You can.
so
Because
that one
you know,
is extracting
the
or
cells they have like
Didn't get what
many
I'm asking.
ligands in the membrane. They have many binding sites. If one GPMV is having binding site and other is having receiver for that recipient.
So your research is all
read about
GPMV.
endocytosis
My
or
research,
something like that?
no, this is only one part of
very
it.
new
I am also
like
with you.
all this stuff.
This is one part of
I
it.
will tell you.
So what kind of things you do?
See, GPMVs, I have just I have just started working like 3 months. Last year I started working, so I have worked in total I have worked for 3-4 months only on that. So what I have done right now is I have extracted them and then I have done shape analysis with them. I have given them like the buffer which I showed you. So now see, we had removed the media and we have added this buffer now. So when we'll be taking the vesicles, so now basically outside the vesicles we'll be having this buffer, right?
Right.
You agree with me? And inside I already told you it is taking part of cytoplasm with it. Okay. So now in this buffer what I have is these two components, and except this I'm making in vesiculation. So these are also there, है ना?
Right.
So this is ion, and these are also too complicated, components. I don't want to, do anything. This is di. So I don't want to do anything with them. This HEPAS, it is not ionic, it is a very safe option to use. So what I wanted to see, we do many experiments on osmolarity change with the with the GPMVs. So I had the GPMV system also, and according to that, if we see, if the theoretical studies also, actually also everyone has talked about that, when we'll keep on adding some components, the rigidity will keep on increasing. We assume this in our mind, and now I'm telling you, if I'm claiming you that GPMVs are the replica of the cells, so it has so many things. It is cholesterol also, it has protein also, it will be very rigid. So I wanted to see how they behave if I do also. I did same amount of osmotic change.
So you're not interested in a very particular component of lipid?
Not yet.
Okay. You're interested in general.
Right now, our first step. This is I can tell you, this is a initial research. So it is the first step to see how they behave in different environments. So I have picked up this component, which is safe for me to change. So with this, it changed the osmolarity by giving a change of 50 millimolar, 50 millimolar, then again 50 millimolar. So 2-3 components I have increased this. So I could.
What is the size range of GPMVs?
It is I think max size what I get is around 20 microns. And 5 to 20 microns, like that. Do you also want to?
Yeah, I would like to see this. So this is cell culture initiative.
Yeah, in which I put it the best solution. These are the small ones, they are merging out. And then also which are already worked out.
So does it need more time of incubation or?
Yeah, actually, and if you leave it at for however long, at least some will be always there which are budding out. But we can see the density in the solution. If we feel it is less, we can give them for more time. Yes. These all are best. So someone who was asking for attached, some we get these structures also. They get attached also to each other. And different types of contrast, these are dark contrast, and some are these also, which are little less contrast, like the UVs, they are visible like UVs also. So dark contrast is, I think, usually because of the cytoplasm.
So what would have happened to the one which is one of this? Less cytoplasm?
Maybe. Maybe. That we can assume that. So this one thing is we have to always move with the hand because if the no stage is compatible with it. So these bigger sizes also. Vesicles have been formed at sea. See, they are moving from. They are all vesicles. There are many. Some are burning out.
Oh yeah.
Yes.
I'm happy with
That's neat. Yeah.
Who's next? HC. Who is HC? You? Oh, you're हाँ वो confluency it depends upon confluency ना कितने है तुम्हारी confluency कम हो गई so that is minus
We have commercial.
cells they are also
So what do you want to use it for?
they are bulging oxygen these are all these are GPMVs. If you leave to bit more, they'll come out. Very little. अब अपने वाले की ले लेना.
मैंने भी यही बोला.
अच्छा.
हाँ दीजिए दीजिए.
अगला एक बार बाहर वाले में देख लीजिए ना तो इसमें दिक्कत हो गया तो तुम्हारी कॉन्फ्रेंस से your formula. Next, PSN.
हाँ महेश
महेश
मुझे PSN की भी
महेश पैर बुला लेना
देखा नहीं? उधर देख उधर दिख जाएगा अभी इसमें दिख गया ना तो सही दिख जाएगा वहाँ देखो yeah. So magnification is I have cited 40x. Now I'm changing focus.
Especially when the speaker.
Like 1.5, 1, like above 1 and it starts vesiculating. Like now we had this 1 hour. So after.
There are not.
So if you keep, you have to give it for more time for that. Like I usually keep 1.5 to 2 hours, but like today we had.
इनके structures देखेंगे.
Into 1.5 hour it gives.
Each of these cell membranes will come out.
Yeah, most of it. How much they can come out. Again, that depends on time. How much you will keep, na?
How do you find the radius after taking
Yeah, from imaging. Like we'll take the images, which are isolated completely. So that I take like phase contrast images, or even I can add like some membrane types. It goes in that membrane type also. They have like anyway this will also be dead after some time. These also dead anyway. When the cell starts blebbing, it is dead. Even if we don't add anything, if the cell is going to die, so it starts blebbing. Next, D3 and DV.
दिगंत भास्कर
नहीं वो भी अलग होगा प्लेस
दोनों बाहर वाला नहीं चला तो देखिए
वो दिखेंगे
बताएंगे
वो सेम ही है सेम ही है माइक्रोस्कोप
हाँ सेम ही है
फिर तो हो जाना
देख रहे हैं एक बार देखेंगे
मैं देखता हूँ
हाँ GPMVs are there. These are the free ones. These are the adhered cells. Okay. So this is the circulating. It might need more circulation, but still like they are. This is the merging out. So these are the free ones. Here they are. See, this is the bigger one. It's merging. So if we incubate it for more time, then we can get this. Yeah, cytoplasm, proteins, lipids, everything. Will be visible, just a little with the little higher contrast. Otherwise, they are same. In visibility, they are same as UVs only. See, there are so many. See, all these are GPMVs which are floating now. All these. That only UVs we have from like 50, 60, 70, 80 micron also we get. And these are like max in my with this method I am getting 20 microns. 5 to 20 microns.
Even we must
M. Who is M? This is the cell. So these are emerging out here. सरफेस से नीचे डिटैच हो गया बट GPMV this is the free one. So yeah, actually these in cells confluency was little less, so we are getting a less number. Like I showed you the other one, the flask, this one. This was having more because the confluency was more in this. These are complete, I guess. बाहर दिख रहा है क्या सर? नहीं दिख रहा। ओह, इसमें बहुत सारे हैं। लैब्स तो इसी को बोलते हैं, वेसिकल्स को जो ये प्लाज्मा वेसिकल्स होते हैं इनको लैब्स बोलते हैं। जो भी सेल्स से ऐसे निकल रहे हैं इस लैब होता है। आजा, यू हैव वेरी गुड। छोटे हैं पर अच्छे हैं। कपड़े में बड़ा दिखाता है। हाँ।
ये एक्सपेक्टेड ऊपर फ्लोट कर रहे हैं सेल नीचे
या ये सेल्स निकल चुका है हाँ सी ये सेल है अटैच हो रखा है और ये देखो ये इसमें से ये निकल रहा है दिस इज आल्सो ये भी निकल रहा है ये निकल के फिर ऐसा हो जाएगा
ये कौन सा सेल है?
जो अडेयर थे अपने जो हम इसमें हैक टू किडनी सेल बहुत सारे नेक्स्ट
भाई मैं निकल
एक में तुम्हारे तो एक में भी काफी ओपन ये सब डेट हो गए जब बस जाना ही कुछ नहीं बनने लग जाएंगे एनिमेटेड विमान कर चलना है ओके See, this image is basically just a visualization of like you have prepared it. अगर तो individual का if you take the image, जो which I have got actually, that you can take and you can like then measure its area, diameter, and also you can do confocal. I have what I have done is I have taken Z scan of that, so I could see the shape. So with the like Z scan, it takes the slices like with the like I have set this interval like 0.5 micron interval. After every 0.5 interval, it takes the slice and then it will form the image. So with that I am like I calculate the volumes of this area.
You need to dye for that.
For confocal I need a dye. So for that I can use this. We have a Nile red. So it is a membrane dye. So it goes wherever it is. But we have to be careful in that because these cells also have a lot of lipid, so we have to separate them because then cells give a large number of intensity. I can write like that also, but the cells they have large number of lipids. So if I'll take a screen, if I'll visualize, then I'll only be able to see cells. They'll suppress the vesicles. So I just separate out that. We can just take the solution from that.
Here इसमें बीच में लिख गए हैं, so it will be in the video.
No problem. It is inverted.
Okay.
होता है
This one.
ऐसा डाउन नहीं होना
Sure.
मेरी यहाँ पे रखो
Mechanical stress, like setup, you would not hear. There's a micropipette especially setup with that also you can do that. Like whatever thing puts stress you can use.
What do you study on how to study
Then see the cells are usually now, if I do it properly, like I deal with regular and this, very less cells will get detached. So they will basically remain attached to the surface. So I can just take out from that. Then I can maybe in phase contrast also I can take the, I'll do the imaging and then I can see its parameters, or I can, if they're flickering, I can tell it. With the flicker we have some programs for that. You can find by integrating surface tension, or I can do an aspiration setup with that. I can pull that and then maybe next सुचिता बाहर दिख गए
वो नहीं उसमें सेल्स दिख रहे हैं और सब बट
उसमें कॉन्ट्रास्ट अच्छा है, दूसरा उसके ऑब्जेक्टिव पे ही ऐसा कुछ है कि ऐसे ही वैसे दिख रहे हैं ओवरऑल।
नया है वो कर दिया है
जैसे कि
बहुत सारे हैं काफी है तुम्हारे वाले से ज़्यादा है
पार्टी में ऑफिस चल
फिर उनको दिखाना है कौन कौन रहेगा कौन कौन रहेगा
हम ये लेक्चर चालू है
हमारे ग्रुप पड़ोसी
नीचे वो
हमारे जल्दी आ गए हाँ ये मजदूर है अभी थोड़ा सा ना एक्चुअली अभी अपन ने जल्दी भी निकाला ना तो टाइम नहीं था वरना लाइक इफ वी कीप फॉर अराउंड तेरी कॉन्फिडेंस ज़्यादा थी ना अगर लाइक 1.5 to 2 आवर्स रखे ना फिर बहुत सारे लगते बट अभी भी ये छोटे हैं अभी इससे बड़े वाले भी निकल रहे हैं हाँ रख सकते बट उसके बाद तो क्या करोगे अभी एक्सपेरिमेंट तो करनी होगी जो लिपिड्स अपन अभी से एक्सट्रैक्ट नहीं ना करेंगे जो ये वेसिकल है ये प्लाज्मा मेम्ब्रेन के लिपिड अपने साथ ही लेके आ रही है प्रोटीन और लिपिड दोनों लेके आ रही है तो अपन जो जैसे यूवी पे एक्सपेरिमेंट्स करते हैं वही इस पे भी कर सकते हैं बस वो क्या हो जाएगा कि वो सिस्टम प्लाज्मा मेम्ब्रेन का हो गया
बट उसमें ये होता है ना कि वो बाइंडिंग साइट है अपने को पता है कि इस इंटरैक्शन की वजह से हो रहा है इसमें
वो चीज़ हाँ वो ये है कि हमें फिर यही स्टडी करना फिर हमें कोई एसेज वगैरह जो होते हैं वो करके देखना पड़ेगा कि इसमें क्या क्या है
ये एक्चुअली सेल के लिपिड्स में क्या मोटिवेशन है
हाँ
जो
एग्जैक्टली राइट जैसे हम लोग जो करते हैं हमारा बेसिकली लाइक जैसे हम तो मेम्ब्रेन प्रॉपर्टीज फिजिकल प्रॉपर्टीज फाइंड करते हैं तो हमारे लिए इट इज लाइक वी डोंट स्पेसिफिकली हम साइट नहीं ढूंढनी अगर हमें सिर्फ बेंडिंग रिजिडिटी फाइंड करनी है सरफेस टेंशन फाइंड करनी है तो हम बस ये बोल सकते हैं ये प्लाज्मा मेम्ब्रेन रेप्लिका है तो हमें एक इंडिविजुअल साइट की जरूरत होती है तो वो स्टडीज हम कर सकते हैं जो उसके लिए तो हमें सिर्फ यही चाहिए कि हमें इमेज है मॉर्फोलॉजी चाहिए हाँ
और डोर डिटेक्ट
हाँ 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 बताया हाँ आपका कौन सा है एक मिनट थोड़ी अच्छी बात वाइट नहीं है एक्चुअली कॉन्ट्रास्ट की वजह से हो रहा है ये भी सेल है जो GPMVs वही होंगे जिनका ये डार्क कॉन्ट्रास्ट these one which are emerging out हैं सारे GPMV ये निकल के फिर ये सोल्यूशन में आ जाएंगे
और जब ये सारे निकल जाएंगे तो ये सेल कैसे हो जाएगी
फिर ये ऐसे ही रहेगी बस ऐसे मर जाएगी ऊपर आ जाएगी
तो मतलब वो कैसे दिखेगी से रप्चरिंग टाइप का
हाँ मतलब ये अभी भी जो आपको दिख रहा है ना दिस काइंड ऑफ मतलब अभी ये तो adhered है, ये तो एक सेल नहीं है ना, ये सारा अलग अलग सेल है, तो थोड़े जैसे ना बड़े, मैंने फाइब्रोब्लास्ट सेल होते हैं वो बड़े होते हैं थोड़े, अगर 60x वगैरह में देखें ना तो हम रप्चरिंग बहुत अच्छे से देख सकते हैं, बट अभी ये 40x में तो मैं थोड़ा हमें इतना ही
मतलब सेल्स डिफरेंट है, मतलब कैसे, जैसे आप बोल रहे हो किडनी सेल है ना,
हाँ
एक ही सेल है ना,
ये एक पूरा एक सेल नहीं है,
अच्छा
दिस माइट बी 2-3 सेल्स अडेयर होके आपस में होंगे,
ओके,
यू नेवर नो हाँ कितने सेल थे, देखिए इतना सारे एक सेल थोड़ी ना है, these are different, ये फ्लक्चुएट भी कर रहा है, तो फ्लक्चुएशन है, you can measure, हाँ तुम्हारे कौन से तुम्हारे कौन से तुमने एंड पे डालना है ना, थोड़ा टाइम कम पास हुआ है, बने तो हैं बट अभी इनको टाइम चाहिए, और इनके छोटे बने हैं, बने तो हैं बट इनके छोटे हैं, बिकॉज अभी तुमने सबसे एंड में दिया ना, इसलिए इसको थोड़ा टाइम चाहिए अभी, क्योंकि अपने को टाइम भी 1 घंटे के अराउंड ही हुआ है, वैसे तो 1.5 आवर मतलब बने हैं, बस थोड़ा टाइम है तो और रख दो, तुमसे चेक कर लेना है ना, तुम्हारी कॉन्फिडेंस भी काफी कम है ना, रख दो कुछ टाइम बने तो है, चेक कर लीजिए पता इंक्यूबेटर में रख देते, हाँ कर लीजिए, छोटे हाँ देख बन रहे हैं, देखो निकल रहे हैं, ये बने वही थोड़ा सा और टाइम चाहिए, हाँ 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 सर